In this video, we will discuss about this microRNA regulation in the Glycan precursor synthesis. So, in brief, how this Glycan formation or Glycan precursor synthesis or glycosylation pathway occurs within the cells is being shown here, and it is a multi-step process. And within this multi-step process, if any of the process get disrupted, then it will uh, results in the change in the final glycosylation profile that is occur within the cells, and these changes are being observed in the tumor cells. So by understanding what are the microRNAs that can target a different glycosylation profile, we can try to correlate those studies in a way that we will get the glycosylation profiles that are similar to the tumor cells, so that we can understand which glycosylation pathways are dysregulated in the tumor cells, and that will provide us a more in-depth understanding of how this mechanism changes in the tumor cells and how the tumor cell functions. So in order to form this Glycan precursor, the one of the important thing that is raw material, or we can say the monosaccharides, are not directly available to the cells. They should be supplied from the extracellular matrix or via food. So these are some of the monosaccharides that are shown here. First one is this D-glucose that is shown here, that enters into the cells. Another one is this glycan, shown here, that enters the cells from here. So whenever this D-glucose enters the cells, after that it get processed, it get transported by this enzyme to the different organelles within the cells, and thereby it this dashed line represent the transportation of this monosaccharides, and this dot line represent the metabolic process, and this arrow line indicates the formation of this Glycan precursor. So in this case, this glucose is added to this 6-phosphate via this metabolic process, and this metabolic process can be taken place can be supported by these enzymes. So let us go through this one of the examples, that is this microRNA. This list of microRNAs can target this transporter enzyme, so that it can inhibit this or reduce the amount of D-glucose within the cells, and this will lead to disrupt of the further glycosylation profiles that are related to this D-glucose. So this is one of the major transporter within the cells for this D-glucose that can be targeted by different microRNAs that are being identified. And similar in the case of this Glycan, what happens is that the transporter enzymes that transport glycan to its final destination within the cells are being targeted by this microRNAs, different microRNAs that are shown here. So this will disrupt the further glycosylation profiles that are being taking place within the endoplasmic reticulum and Golgi. So these are two examples that I have shown you, and there are other examples are also that is shown here. For example, this transportation of this UDP GalNAc, or in other terms, this formation of from this Glycnac 1 phosphate to UDP GalNAc is been done by UAP, and this UAP enzyme can be inhibited by this microRNA 244-5P, and this will leads to less amount of or inefficient supply of UDP GalNAc within the cells. And another examples that affects the fucosylation within the cell surface is this GDP fucose is converted into GDP4 oxodeoxymanose with the help of this microRNA 125. So this microRNA 125 will disrupt this reaction, as it will inhibit this GFUS enzyme that helps in the this reaction of converting GDP fucose to GDP4 6 deoxymanose. So these are some of the microRNAs that are being identified that disrupts these glycosylation profiles. If we can study these changes in this cell system, changes in the glycosylation profiles in this cell system, and somehow able to produce the glycosylation profile that is similar to the glycosylation profile that has been found in the tumor cells, then we can understand what are the enzymes that get disrupted in the tumor cells by comparing the tumor cell glycosylation profile and this well controlled model of the cell system that produce the similar glycosylation profile as that of tumor cells. So we will from this control system we will have an idea about what are the changes that are occurred in the cells that leads to the glycosylation profiles that is similar to the tumor cells, and then we can and then we have the more in-depth understanding about how this mechanism how this cell signaling mechanism in the tumor cells changes, so that we can work on reversing those mechanism within the cells and try to convert those tumor cells into the healthy cells. This will leads to an approach where instead of killing the tumor cells, we can reverse the tumor cells, so that we will have we can reverse the cancer-like diseases into the normal healthy cells, or we can reverse the cancer-like disease patients to the normal patients by reversing these tumor cells into the normal healthy cells. So this is now something a correct statement. This is now something like a correct statement. If you need to understand more about this microRNA regulation in the Glycan precursor synthesis, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.